you how to do that as well. Reproducibility is a real key in translating this therapy. So when you isolate one stem cell one day and come back next year and isolate another stem cell using the same protocol, are those two populations the same? And for regulatory issues, you have to prove that they are the same. And so we've been working on that with a number of different molecular biology techniques. Cost effectiveness is also an issue going forward. iPS cells, as you heard from the first talk, can be used to generate photoreceptors. It's really long and expensive to do that. The reagents that are required to do the retinal protocol differentiation step are very expensive. And again, with human cells, it takes about half a year to generate photoreceptors. So with the cells that we're using here, because they're already committed to the retinal phenotype, it takes 7 days in culture to differentiate the human cells into photoreceptors. And that's a big difference. And you don't need to doesn't require any exogenous factors, just a good substrate. Okay, so there are lots of places, of course, to get self-renewing pluripotent cells. Again, we're going to hear about all of them today. I'm going to talk to you about tissue-specific cells isolated from the CNS, in this case, the retina. So this cartoon highlights a couple of the issues facing us when we're trying to transplant new cells into the retina. The real key is when to do it. Not when from the point of view of disease progression is a whole other question. Let's go a step back and figure out when do you transplant the donor cells in the stage of differentiation. If you're playing with commitment versus plasticity. Early cells might be very plastic, but might not make the kind of cells you need. If you pre-differentiate them into mature photoreceptors, we already know they're not going to integrate and not going to make connections. So somewhere in the middle is the optimal. And we're trying to figure out what that is. The work from the London group, Robin Ali's group, would indicate that the stage that says post-mitotic photoreceptor precursors here is the ideal stage, at least for mouse-to-mouse transplants. So we're isolating cells and growing them as lineage-restricted progenitors, but they're still proliferating, okay? And the question is, do they still make photoreceptors? And I hope I convince you that they do still make photoreceptors after transplantation. So this is what the human retina looks like at the stage that we're isolating. This is about 16 weeks of gestation. The retina at this stage has some committed cells, some cones, early-born cones out here, some early-born ganglion cells here, but most of the retina is neuroblastic at this stage. And if you stain it for markers of pluripotent cells, or at least of proliferative cells, SOX2 here, and Ki67, we see that there's abundant pluripotency and proliferation taking place, but very little commitment to specific cell types. Again, we see a few early-born cones expressing recovery, but no rods have been born yet at this stage. And we're not sure what this recoverant staining here is in the ganglion cell layer, but we don't think they're photoreceptors. So what we've been doing is trying to characterize the phenotype of these cells using a bunch of different approaches. One of them is flow cytometry, looking for surface markers. And here we have different isolations and different passages. So these are two different isolations. We're calling them 86 and 89. And they're showing, at least for 86, passage 9 and passage 14. And looking at the expression of some of these interesting markers that we're interested in. SOX2 is expressed by over 95% of these cells, and that maintains after passaging. PAX6 is a little more variable. It's around 50%, but it does change a little bit. It just seems to be a little fluctuation. There's no consistency to it, but somewhere between 45 and 60% of the cells are SOX2 positive. CD73, a marker that's been used to isolate subpopulations of photoreceptor precursor cells, are expressed in high levels by these cells, regardless of passage. Interestingly, we don't see any CD133, typically a marker for neural stem cells. And these are neural stem cells from the eye. So we don't see that, and that's interesting. We also don't see, for instance, A2B5, a marker for developing glial lineage. And so these cells are not very good at making Mueller cells or astrocytes. So one of the things that we've worked on is, again, how to grow these cells into enough cells to use, to characterize them and to transplant them. And so in the old days, typical incubator conditions, this is what happens. Senescence at about 45 days. not enough cells are generated. You can't continually isolate these cells. So what we had to do is figure out a way to expand them, and the work of Marie Chetta in other stem cells has shown that low oxygen, 3%, 5% oxygen, leads to increase in pluripotency and increase in proliferative capacity. And now the limits are off of these cells. We can now grow them for long periods of time without them changing or senescing, up to 20 passages, which gives us trillions and trillions of cells. This is the banking strategy that we have. So we have banked these cells, 3 different cell lines under GMP conditions with seed banks and working banks. This has all been done with a collaborator in Philadelphia, a WuXi laboratory outside of Philly. And they've been doing all these GMP isolations. So they use our technique to isolate these cells and do it under GMP conditions. So we have enough cells. That's not the problem. The question is, what do these cells become when you differentiate them? So we're looking at PCR, immunohistochemistry, and flow cytometry methods of confirming whether these cells have the capacity to make photoreceptors or not. And we use an in-culture assay I won't tell you much about. I'll tell you nothing about it. It's a polymer substrate called polycaprolactone, coated with fibronectin. Something magical about the stiffness or the Young's modulus, no relation to me, of this polymer substrate induces very rapid differentiation of these cells and other cell types of the neural lineage into whatever cells they're going to become. So can they make photoreceptors? They can. So this is showing an explant assay, and I think I'll make it. Grafted cells are green, added to a retina, now stained for photoreceptor markers. This is rhodopsin. And what we're seeing here is that the outer edges of these cells are forming what look to be like outer segments. And I think I'll show you some images of these cells stained for RON1, peripherin, other markers of outer segments. But here they line up in the outer nuclear layer and start making rhodopsin. If we transplant them into a mouse, we can see that they also, here's an OCT image, really cool new OCT machine we have at the Institute. You can see the bolus in the subretinal space. And then we go in and label these human cells for human mitochondrial markers, and again, see them lining up in the outer nuclear layer where we want them to be. So they migrate to the right place, and they also make photoreceptors. Here we're seeing, again, rhodopsin staining. So all of this is a rhodopsin knockout animal. So this animal has no rhodopsin. That's why the outer nuclear layer is not stained positive. These orange cells are donor cells that are making rhodopsin and integrating in the outer nuclear layer. I want to tell you it's a real challenge to do human studies in mice recipients. The cells don't survive very long, and some of our future work is going to make use of immunodeficient mouse strains that have retinal-degenerative backgrounds. These have been made by a former student of mine, Bud Tucker, and there we can really study what these cells can become over time. Just another image of transplanted cells expressing rod markers after transplantation. We've done some work in RCS rats. These cells also rescue diseased photoreceptors in RCS rats. Not surprisingly, they make lots of growth factors. And they do it anatomically as well. In pig-to-pig studies, we've seen that these cells can also make photoreceptors. So this is a bolus of cells in the subretinal space. These are all new photoreceptors sending synaptic processes into the appropriate plexiform layers here. Okay, so just to summarize, the retinal progenitor cells that I've described to you can be isolated from embryonic retinal tissue, including humans. We can expand them into many trillions of cells in a dish. And like iPS cells and ES cells, they make rods and cones when differentiated, but unlike these cell types, we think they're very safe and unlikely to proliferate after transplantation. Thanks very much.
I just want to thank the Hope for Vision group very much, because they gave us $25,000 several years ago, and I think probably Betty Feeney had something to do with that, which allowed us to do the first work in animals with the vectors that I'm going to tell you about today. I'm at Florida Atlantic University, which is a state university north of here, and we don't have things like lasers, and it was very important in the early days of testing our vectors to be able to use an animal model, and we used an animal model at that time using lasers so that we would have a model for CMV. But what I'm going to talk about today is actually the model that we use for diabetic retinopathy. So there are two diseases that are well known that have abnormal angiogenesis in the adult. One is AMD, and the other one is diabetic retinopathy. And I'm going to tell you today about our results in an animal model of diabetic retinopathy. So as I mentioned, I must say that you're going to be one of the best informed lay audience that I've ever spoken to. So I'm going to go over it without a lot of detail, and of course I do have slides with details if that's important too. So in diabetic retinopathy, what happens, the hyperglycemia leads to vessel loss. And the vessel loss gives you an area of hypoxia ischemia, and that causes neovascularization to occur in this adult eye. And of course we know that when you have adult angiogenesis, you end up with vessels that are very abnormal, and they break easily, and you end up with blood in the eye, and eventually you end up with loss of vision. The photoreceptors degenerate. So this would be what we normally see is there is a blood vessel supply in the choroid, and then there's some inner vascular supply that's coming here from the central artery, the ophthalmic artery, and the inner retina is supplied in this region. But what happens in diabetic retinopathy is you have this area of hypoxia, and that eventually leads to this massive growth of blood vessels into the vitreous. You also end up with epiretinal membranes, and it's a very nasty, deleterious situation. So what we did in trying to devise a gene therapy method was to try to optimize some of the situation that occurs in this pathological state. So we were interested in which cells we would target. We thought it was important to include a promoter, so we would target a cell type that could be turned on. And we wanted to use a rescue gene, so we chose to use endostatin, which has been shown by several other labs to stop neovascularization that occurs in these animal models. And we wanted to be able to regulate the levels of production of our rescue gene. And we did that by using a hypoxia responsive element. So what our hope is that our strategy is that we use something called an HRE. It's a set of genes that comes actually, in the beginning, it came from the erythropoietin gene, which causes the cell to be able to sense changes in hypoxia. We add a few other pieces of DNA to that. We use a cell-specific promoter. Then we have our gene of interest. And what happens is, under normal conditions, if this vector gets into a cell, a silencer is being used because it's normal oxygen concentration. However, when hypoxia happens, HIF1 binds to this hypoxia responsive element, and HIF1 is hypoxia inducible factor. And now what happens is we have our vector is turned on, and we have the production of the gene of interest, which in our case is endostatin. And that should lead to the reduction in those abnormal vessels. So in trying to decide what cell to use, we chose the Mueller cell because it extends from the region out in the region of the inner segments all the way to the inner limiting membrane. And again, those abnormal vessels are all down in this region here. So we thought a Mueller cell would be a great target to produce that endostatin. And this is just a picture of a mouse retina with lots of GFAP. GFAP is the promoter that we used. It's found in abundance in Mueller cells. And our paper that we have on this project was first published, and what it showed is that we could take this vector and we could take Mueller cells, we could get the vector into Mueller cells, and we could then put the Mueller cells in a normoxic condition and a hypoxia condition and show that we could turn on the vector and have GFP, the reporter gene, produced. We did a little bit of animal work also during this study. This is primarily the work of our graduate student, Manis Biswal, at the time. Manis has now moved on and has moved up the state and is in Gainesville working with Al Lewin. So we wanted to test the vector that has the GFAP promoter, and we wanted to show that it could stop neovascularization in an animal model. So we used here an unregulated vector, if you will. And we also used our experimental vector, which has these hypoxia responsive elements. So if these two vectors get in the cell, this would be turned on all the time. This would only be turned on if there was hypoxia. So we considered as a control, the opposite eye had either PBS or it had just a naked capsid to be sure that our results were not due to something about the AAV, and we used AAV2, single-stranded AAV2. So I'm going to talk a little bit about the morphological analysis and the biochemical analysis. We used as a model that's probably very familiar to everybody in this audience, and that's the oxygen-induced retinopathy mouse model. You take young mice with their mother, and they're at P7, you put them in high oxygen, you take them out at P12, and you leave them in room air, and you get this huge growth in vascular tufts in the region of the peripheral retina, and you have vascular degeneration in this area of the central retina. However, if you leave them long enough in room air, the vessels will actually recede. So the period of hypoxia is important, and we decided to do our intravitreal injections here. And then we did our data analysis at P17. So this is how we analyze the data. We make a whole mound after we do a vascular perfusion of most of my life was spent with peanut agglutinin, but now I'm into labeling blood vessels. So this is actually tomato lectin, which wasn't even around when I did the earlier lectin work. Anyway, we perfuse with our lectin, and this would be the regulated vector, the retina from an animal that received the regulated vector. This is the other eye, which has PBS, and you can see that there's this huge growth of blood vessels and then this avascular area here. However, when we look at our experimental situation, we see that as in a normal situation, this is more vascularized here in the center, and the periphery, we don't have these huge globs of vessels anymore, but we have a nice vascularization. So the summary of our morphological data is that we had a 90% reduction in the neovascular area in the periphery, and then a 90% of the reduction in the avascular area. So that avascular area became vascularized. So I just am going to tell you the summary of our biochemical data. We were able to show that there is normoxic silencing. The regulated vector does not show endostatin expression in the retinas of mice in room air. We showed a hypoxic induction when we give our treatment by doing ELISA and Western blotting to show expression of our flag-tagged endostatin, which is in the GFAP vector at P17 in those mice that were in this OIR model. And we also saw that our vector did have, we showed that it had exogenous expression of endostatin by either vector was able to reduce the VEGF level in this OIR model mouse retina. So in conclusion, we specifically designed hypoxia-regulated glial cell-specific vectors that inhibited neovascularization in an animal model of diabetic retinopathy. We think the significance of our work is that we have a regulated switch for gene therapy. So we hope that our vector is turned on at the right time, in the right location, and at the right level. And this is what we think groundwork for gene therapy to treat neovascular disease in its initial phases. What we're hoping to do now is to inject the vector, to wait 3 or 4 months and see if we can turn it on after the vector has been in the eye for a number of months. And these are my collaborators at FAU. And the other folks gave us pieces of DNA. GFAP promoter Keith Webster gave us the initial HRE, and Douglas McCarthy gave us the single-stranded AAV. Thank you.
Thanks for inviting me. This morning I'd like to talk about some optogenetic projects that we're doing in my lab at Berkeley in collaboration with the Isikoff lab, and we'll also talk a little about some other optogenetic approaches that Rich Kramer will talk about in a minute after me. So the idea in general for these optogenetic therapies is that this anatomical organization of the retina in the wild type state or the normal state is like this, where we have the layers of the retina photoreceptors to the inner retinal. But in most cases of advanced retinal diseases, what happens is that the mutations cause the photoreceptors to be lost, and in that situation, the only cells that respond to light have died. However, it looks like that these cells, the bipolar cells, and the downstream cells from that, the interneurons of the ganglion cells, are still connected and probably still functional for very long periods of time, maybe years, after the patient has lost vision by the fact that they have no more rods and cones. So many different groups have decided to try to augment or design a genetically encoded way to restore vision to people that no longer have photoreceptors by adding an optical switch. And Rich will talk about one, Alan will talk about another one, I'll talk about one in the other part of this talk. But generally, these switches can be targeted to different downstream signaling parts of the retina. So Botan Raska and Josai Sahel are interested in adding optical switches to surviving cone inner segments in patients that no longer have rods or cone outer segments. Other groups are using switches targeted to bipolar cells, and other groups are targeting the most downstream, the retinal ganglion cells. And the thought is that the amount of restoration of function may be proportional to which target you use. So obviously the ones that are closest to the top, which are the cone photoreceptors, probably retain the most of the circuit, the center surrounding the edge detection. And then other patients who probably don't have surviving photoreceptors would get more restoration from bipolar cells, but less than if they had cone as their restorated cell. And then finally, there's some patients that probably only have surviving ganglion cells. So there are many different optical switches that groups have decided to use. The naturally occurring switches that have been used are channelrhodopsin and halorhodopsin. Rich will talk about engineered chemicals, AAQ, and other derivatives of this that are azobenzene derivatives that modulate endogenous ion channels in the retina. We published 2 years ago a glutamate receptor that we genetically transferred to retinal ganglion cells. This is a summary slide that's prepared by the staff of the Foundation Fighting Blindness, summarizing the results in the literature. And what I want to call your attention to is that all these different approaches, whether they're the glutamate receptor, channelrhodopsin, halorhodopsin, or melanopsin, and regardless of which target you have, whether it's ganglion cells, inner segments, or bipolar cells, all of them have a similar characteristic in the fact that they're significantly much less sensitive than normal wild-type human vision. So they all operate in the upper part of the cone range. It's probably easiest to see here. They're operating in a light sensitivity that's about the level of a bright June day or the Miami Beach today. And they don't adapt because all of these are one component, if you think, systems in which the light sensor and the ion channel are the same. So they have very insensitive, relatively insensitive, and also this range is only up in the high end of the photopic range. One of the reasons we think that this is true is the fact that if you're moving the photo switch, whether it's channelrhodopsin or one of the other switches, to one of the downstream cells, whether it's bipolar cells or ganglion cells, this is the anatomy of the cell you're trying to replace, which are photoreceptors have this huge antenna, if you will, of membrane of outer segment. And this is packed shoulder to shoulder with the light-sensitive compound opsin. So in the case of these other cells, it's basically just the same as the inner segment, whether it's a bipolar or a ganglion cell. The surface area available for adding these photo switches is much, much less. So one of the thoughts of a student in the lab that's shared between Udi Isikoff's lab and mine, Benji Gaub, is to separate the light sensor from the ion channel. And that's what photoreceptors do, and that they have the light sensor as rhodopsin, then they have the G protein cascade, then they finally have the ion channel in the plasma membrane. And so it may be well that if you separate the light sensor from the effector, that gives you back some of the adaptation and perhaps some of the amplification that you see in photoreceptors that you don't see in these other approaches. So what Benji thought to do is to add a GPCR. In this case, there's a metabotropic glutamate receptor 6 that naturally occur in bipolar cells, and these receptors actually have this separation between the effector and the ion channel. So the idea is, can we hijack a G protein link cascade in a downstream cell, like a bipolar cell, and get back some of the amplification and the adaptation? So the idea is to design vectors that will transfer new switches to on bipolar cells and use that as a target for expression of GPCRs. And so one of the things we thought is the first example of that is to use rhodopsin. And so we designed an AAV vector with a promoter we got from Botan Raska and Connie Sepko. It's the GRIM6 promoter. It's actually 4 copies of that driving a GPCR. Here's the expression of that. It's very highly efficient for transfecting on bipolar cells. And the functional assessment is in a multi-electrode array. So we lay the retina down and record from ganglion cells, but these are being driven by optical switches that are in on bipolar cells. So what, in the first set of experiments Benji was able to show is that rhodopsin transferred to on bipolar cells drives light sensitivity in ganglion cells. And here's a raster plot. Each one of these traces, here's the light pulses. Each one of you see the activity goes way up, and this is about 30 different ganglion cells being recorded simultaneously, being driven by the on bipolar cells. Here's another example, and what he's comparing here is wild-type retina sensitivity compared to rhodopsin and channel rhodopsin in bipolar cells of RD10 mice that no longer have surviving photoreceptors. So what he's able to show is that you get significantly more amplification. Here's the sensitivity of rhodopsin compared to either channel rhodopsin or the glutamate receptor expressed at the same level in the same cell. So it appears to be that there is some amplification and hopefully some adaptation by hijacking and separating the light sensor from the effector. So in the context of human vision, what is this? Here's the measurements I showed you earlier, sunny day indirect light. This is channel rhodopsin in the on bipolar, the glutamate receptor. The measurement that Benji has made using rhodopsin in the on bipolar is about weak office lighting. So it's about 100-fold more sensitive by just separating the two into a cascade, if you will, than what's possible by using a one component expressed at the same level in the same cell. So in summary of this mouse work, we've identified at least one naturally occurring GPCR that couples to an endogenous ion channel in on bipolar cells. And we've made virus vectors that efficiently express that in on bipolar cells. And we think that they can reanimate the blind retina and enhance the sensitivity and maybe add some adaptation compared to other systems that have a single ion channel that's also the sensor. So we're starting to move on to test this in larger animals. And this is a collaboration with William Beltran and Gus Aguirre at Penn. And so they've started to make animal models with behavior that you can test only in dogs and you can't test in rodents. And they've looked at 4 different dog models to test this. And what they've done is they've built obstacle courses that they can test the vision for optogenetic switches in dogs. And so these are the protocols. I don't really have time to show them now, but this is the example of it. It's a large Y maze, and the dog enters the maze here, and then it has an optical task that it can see here. That's a stimulus, and it makes a decision whether to go right or left. And then finally, as an exit, which is sort of a doggy door. And what's surprising when you see that is the dogs just like really love to do the task. They run through the maze, they look at the target, they go right or left, they come out here, and then they run around the front again and ask to do it again. So they've built this optogenetic Y maze for testing whether or not it's the single component that Rich will talk about in a minute, AAQ or FENAC, or the genetically encoded components that I've talked about in the mouse work. And they're starting to test that in the dog animals. And so this is an example of how you do it. The dog comes to this decision point. You test whether or not they can see the brightness and the acuity by projecting it on a screen. And the dog makes a decision. One of the challenges that they found is that most of the dog models are really not that blind. So they have to induce light damage. The one that looks like the best example is the rhodopsin mutation, T4R, where they can induce light damage and then deliver the optogenetic tool. The last thing I'll talk about quickly is that we're also collaborating with Bill Merrigan and Dave Williams at Rochester to do these kinds of experiments in macaque. And this is the pattern that we see with the same vector I showed you earlier. However, in the macaque, what you see is because the interlimiting membrane is a barrier for the virus, so you inject in the vitreous, what you see is expression just around the macula. And the virus doesn't seem to have very good penetration outside the macula. However, this still is encouraging because the number of cells that you see in the macaque is hundreds of thousands. These are ganglion cells that are transfected. So that may still subserve useful vision. So what Rochester's trying to do is to make a macaque model of blindness, which is challenging because there isn't any genetically available macaque model. So they're using a laser to ablate small areas of photoreceptors and then do psychophysics. So what they'll do is they'll test the cortical recordings by driving the system with the photoreceptors, then ablate the photoreceptors, and then drive the system either with vectors expressing the photoswitch in the on-bipolar cells or in the ganglion cells, and then see how the cortex responds in the same animal compared to driving it downstream from bipolar cells from driving it in photoreceptors. So they're getting to the point where they have a macaque model where they can do psychophysics, and they can do that with optical switches, and they can test the difference between photoreceptor-driven vision and downstream driven vision, whether it's from bipolars or ganglion cells. I'd like to stop there and thank all the people that do the lab work, the grad students and postdocs in my lab. Particularly, this is Benji Galb's experiment in collaboration with Denise Salcara and Natalia Caporali. The dog work, as I said, is done with Gus and Bill and Penn. Dave Schaefer helped us design the virus vectors, and the guys in Rochester designed the macaque work. Thank you very much.
We're narrowing in on sort of a one vector fits all cone applications, as you'll see. Okay, so first question, why target cones? Well, this audience, I don't need to describe that, but if you knew nothing about cones, but you saw the vasculature, the retinal vasculature of a primate, you would say, well, there's something funny going on in the middle. And that's the avascular region. Is this not on? That's the avascular region that is over the cones and over the foveal cones. And of course, it's avascular, so you have the highest acuity in the foveal cones. A reason for studying cones. And this just is a little bit better anatomical view of the retina here, human retina, and you can see that if you look down here, a plot of cone density versus rod density right in the middle, right in the foveola, is the highest cone density, and that corresponds to this region here in the histological section. So this is the region that we're really trying to target because it's responsible for our highest acuity vision, color vision, face recognition, reading. Okay, what cone diseases are important to think about treatment for? The criteria are really pretty obvious again. We want a gene that targets foveal cones. The genetics needs to be well established. It should have a serious human phenotype, as it should have a blinding phenotype. We'll talk about one phenotype in cones that isn't a serious disease, but there's a reason for that. And the animal models for that specific cone disease need to mimic as much as they can the human disease. And obviously there needs to be a clear clinical endpoint if you really want to get into a clinical trial with cone-targeted gene therapy. So let's look at the genes that might be involved. This is, of course, a cartoon of phototransduction. And what I want to do is highlight the cone genes that can cause foveal disease. There are three that cause achromatopsia. Achromatopsia is a loss of all cone function. And that's a serious blinding disease. Patients basically have no central acuity, see with their extrafoveal photoreceptors, rods. And those genes include a minor form of achromatopsia, the alpha subunit of transducin, GNAT2 gene, and the two major forms of achromatopsia, the A3 and the B3 subunits of the cation channel. That's sort of the final step in hyperpolarization after photon absorption. So there's three genes there, and there's two more, the red and green opsin genes. If one of them is lost, it will lead to X-linked color blindness, red or green, which is the non-serious cone disease, and then a much more serious disease, blue cone monochromacy, where both red and green opsins are lost, and so you really still have no foveal function because there's no blue cones in the fovea. So this is maybe not quite as serious as achromatopsia, but it really falls into that class as well. So just a bit about delivering the vectors. At least up until a year or so ago, it was thought that you had to go subretinal with your viral vector. And in the case of anything that's not a primate, the cones are kind of spaced out amongst the rods, and so a subretinal vector needs to deliver its payload to the cones under this circumstance. So we're going to have an AAV serotype, and an AAV5 serotype, as we have found quite a few years ago now, is very efficient at transducing rods and cones. We need a cone-specific promoter, and then we'll have a specific cone cDNA for that disease that we're trying to treat. For most of what I'll say, almost all of it, we're going to be using